And I think it's why our mission today is so critical. The sources we've synthesized reveal what is basically the ultimate survival manual, what we're calling the 2025 Oncological Toolbox.
Mhm.
This is a catalog of dozens of interconnected pathways, radical metabolic changes,
Oh.
epigenetic hacks,
Hmm.
and complex physical manipulations the disease uses to guarantee its longevity, its ability to spread, and its terrifying invisibility to our immune system.
And we have moved far, far beyond just looking for well-known markers, like, say, a single BRAF mutation.
Hmm.
Our goal here is to synthesize these dense, really technical mechanisms. I mean, from how cancer hijacks your own body's infrastructure
Yeah,
to how it actually controls the bacteria in your gut,
right.
and extract clear, understandable insights for you, the listener. By the end of this deep dive, you won't just know the names of the pathways. You'll understand the sheer layered complexity of the fight, and crucially, how scientists are now learning to target the wiring that connects these pathways, instead of just the isolated hubs.
Okay, so let's unpack this. We have to start with the foundational infrastructure. If cancer is a car, the first thing it does is permanently wedge down the accelerator pedal. So this is section one, ensuring nonstop growth and, well, literal immortality, regardless of the body's safety checks.
Yeah, so when we talk about core infrastructure, the first thing cancer needs is that constant, just unrelenting signal to divide. And the classic pathway for this is the MAPKIRK pathway.
Right, the one we always hear about with BRAF or CRAF mutations. So for the listener, why are these mutations so effective at kicking off that growth surge?
So the MAPKIRK pathway is, it's essentially the cell's main communication line for growth and proliferation, and is triggered by signals from outside the cell.
Okay.
And RAF is a key protein kinase, sort of right in the middle of this line. When it's mutated, particularly in something like melanoma, it becomes constitutively active. I mean, it's permanently switched on.
Permanently on. It doesn't need a signal anymore.
It doesn't need the external signal anymore. It just screams, grow, at full volume all the time. And that forces this rapid unchecked cell division, which is, the very definition of initial tumor formation.
That makes perfect sense for the initial surge. But what the sources make explicitly clear is that cancer never, ever relies on just one switch. It builds in massive redundancy right from the start.
Oh, absolutely.
And that leads us to the critical backup plan, RAS mutations.
Exactly. The RAF mechanism is that critical initial surge, but RAS, and we're talking specifically KRAS, NRAS, and HRAS, is the essential backup plan. Or actually, often it's the primary driver itself, particularly in these extremely difficult to treat cancers like pancreatic and colon cancer.
So it's another way to get to the same result.
It's a parallel highway leading to the same destination. RAS is an upstream regulator that also feeds directly into that same MPK pathway. So if you target RAS,
RAS just picks up the slack.
it picks up the slack and keeps the growth signal screaming just as loud.
Okay.
And it's this redundancy that explains why targeting one component, like BRAF in the early days of drug development,
Yep.
it led to some initial success, but then very rapid relapse.
You've silenced the trigger, but the whole network is still live.
The network is live.
Mm.
And the redundancy doesn't even end internally. As the sources note, cancer also installs what they cleverly call extra grow buttons right on the cell surface.
And these would be the amplifications of receptors like EGFR, MET, and HER2.
Yes, exactly. These are receptors that are meant to capture growth factors from the outside world. But when cancer overexpresses them or mutates them, they become permanently dimerized or, paired up, and they're just constantly signaling for division and proliferation.
Regardless of whether a growth factor is even there.
It doesn't matter. They are over-engineered switches that are perpetually on, bypassing all the normal regulatory steps entirely. The first lesson is just survival through redundancy.
Okay, so if the accelerator pedal is permanently floored, the second critical thing the cell has to do is remove the brakes. This brings us to cell cycle control and, of course, the evasion of apoptosis or programmed cell death.
And this is maybe the most fundamental survival hack. If a cell is dividing that rapidly, it's going to accumulate errors. It's going to get DNA damage.
Of course.
And normally, the cell has these built-in mechanisms to recognize that damage, stop the cycle, and either fix it or commit suicide.
And this is where the legendary P53 comes in, the guardian of the genome.
Absolutely essential. P553 is the most famous tumor suppressor in history for a reason. It was the master safety officer. When DNA damage is detected, P53 rushes in, stops the cell cycle, and allows time for repair.
And if it's too bad?
If the damage is too severe, P53 forces the cell into apoptosis. But in over half of all human cancers, P53 is either mutated or deleted, or sometimes it's suppressed by another protein called MDM2, When p53 is gone or disabled, those crucial DNA damage breaks are completely removed.
So the cell just keeps going.
The cell keeps proliferating, carrying all its dangerous mutations forward with no consequences.
It's not just breaking the rules, it's literally shredding the rule book for DNA stability. And to speed up division even more, cancer also hijacks the core mechanism that forces cells through their checkpoints, the pairing of CDK46 and cyclin D.
Right. You can think of CDK46 and cyclin D as the required toll booth operators for the cell cycle. They manage the transition into the S phase, which is the point of no return where a cell commits to replicating its DNA and dividing.
So cancer just overproduces them.
It overexpresses this partnership, essentially making the cell blow through these critical checkpoints at maximum speed. Division happens way faster, regardless of readiness or any external signals. And this rapid acceleration is why CDK46 inhibitors are so critical in certain breast cancers. They're designed specifically to reapply those brakes.
To force the cell to slow down or just stop.
Exactly.
So, okay, we have accelerated division and we've removed the death brakes. But cancer still has to actively fight against signals that tell it to die, which brings us to two master survival pathways, NFP and PI3K-AKT-mTOR.
These are the heavy hitters of cellular survival. Let's start with NFE. It's a central player in inflammation, which we'll definitely get to later, but its role here is mainly anti-apoptosis. It actively tells the cell, do not die.
It's promoting survival under stress.
Exactly. And at the same time, you have the PI3K-AKT-mTOR pathway. This pathway is like the primary switch for survival. It turns on processes that promote protein synthesis and growth, and it actively blocks any pro-death signals. And crucially, it dramatically boosts the cell's metabolism, what the sources call metabolism on steroids.
So it's making sure it has the fuel to survive the stress and keep expanding.
It's the core signal for growth, survival, and metabolic acceleration, all wrapped into one powerful package.
So if that system is the engine, the final piece of this whole infrastructure is achieving true biological immortality. We're talking about bypassing the natural limit on how many times a cell can divide, and that's where telomeres and TERT come in.
It is the cellular fountain of youth. Normal, healthy cells have these caps on the ends of their chromosomes called telomeres.
Right, and they get shorter every time the cell divides.
They get shorter every time. And once they shorten past a critical point, the cell enters senescence and stops dividing. That's just cellular aging. So cancer achieves immortality by activating telomerase, often through these TERT promoter mutations. This enzyme rebuilds the ends of the chromosomes, allowing for endless, indefinite replication cycles.
The cancer cell never hits retirement age. It can divide forever.
Forever. And to power this endless immortal division, you need a single aggressive master regulator that just drives everything to maximum capacity. And that's MYC.
The super oncogene.
It's the super oncogene, yeah.
Yeah.
It's frequently amplified in many aggressive tumors. It's not responsible for just one pathway. It's a global command that pushes the entire system growth, metabolism, replication, telomere maintenance into overdrive. Amplification of MYC means it literally cranks every growth gene to 11.
Wow.
When MYC is highly active, it defines aggressive, rapid, relentless proliferation, which makes it one of the most difficult targets in all of oncology because it's just so pervasive in its control.
So to sum up this first section, cancer's core infrastructure is this redundant network of growth signals, RAF, RAS, EGFR, a dismantling of the brakes, P53, CDK46, an active suppression of death signals, NFTPI3K, and then a guarantee of eternal life, telomerase MYC. That is a perfect, self-sustaining system ready for the next stage invasion.
Yes. Once cancer has secured that immortality and nonstop growth, the next stage of the survival manual is escaping the primary site and invading distant tissues, metastasis. And this requires the cancer cell to transform into a shapeshifter and a crawler.
This sounds like some kind of biological Houdini act. I mean, how does a fixed cell, one that's stuck to its neighbors, suddenly gain the ability to migrate and dissolve tissue?
The transformation process is orchestrated by a genetic program, and it's known as the epithelial mesenchymal transition, or EMT. So epithelial cells, like the ones lining an organ, say the breast or the colon, they're fixed, they're organized, they're stuck tightly to their neighbors. They're easy targets because they're predictable. To become truly invasive, cancer cells have to shed this organized, fixed nature and transform into these highly mobile, spindle-shaped cells. The sources accurately call them crawling ninja cells.
Crawling ninja cells? So what are the molecular switches that flip this identity crisis, that turn a predictable cell into a ninja?
We're talking about central transcription factors, the master conductors, things like TWIST, SNAIL, and ZEB1. When these get activated, they orchestrate a complete shutdown of the proteins that maintain cell adhesion, like E-cadherin.
So they cut the ropes holding them in place.
They cut the ropes, and at the same time, they activate proteins that give the cell fluidity, mobility, and these invasive properties like N-cadherin and vimentin. It's a wholesale change in cell architecture that allows them to detach, elongate, and start crawling.
You know, I find the role of TGFO in this process particularly unsettling. We learn about it early on as a tumor suppressor, but here it's actually implicated in driving metastasis. It feels like the ultimate molecular betrayal.
It truly is. In the early stages of tumor formation, TGFO acts as a potent tumor suppressor. It tries to stop cell division and encourage differentiation. It's an early break.
Okay.
But as the tumor progresses and accumulates mutations, the cells become deaf to that stop signal. And instead of suppressing, they hijack the TGFO signaling pathway. It flips its function and becomes a powerful promoter of EMT, of motility, of metastasis.
So it goes from being the cell's protector to the cancer's getaway driver.
That's a perfect way to put it. It literally helps the tumor escape its confines and orchestrates that transition to the ninja cell state.
That transition is just critical. Now, once the cell is ready to move, we need to understand the mechanism of movement itself. This isn't passive floating. This is active brute force crawling. And the sources highlight a specific motility machine, the ARP23 complex.
The actin-related protein 23 complex, or ARP23, is the cell's heavy-duty motility engine. Its entire job is to rapidly generate the force needed for the cell to push itself forward into the extracellular matrix.
And it does this by messing with actin filaments.
By rapidly nucleating and branching them, yeah.
The sources used this term fake podding to describe the crawl. Can you explain that?
Sure. In normal cell movement, you often need some kind of external scaffolding or the cellular matrix, the ECM, to provide anchors. But ARP23 allows for self-generated propulsion. It pushes this branched network of actin forward, creating structures that act like miniature feet or tank treads. Those are the fake pods.
So it doesn't rely on the usual support.
Not at all. It generates the push force internally, and this allows it to move through these dense, constrained environments like a miniature cellular tank, often using brute force.
And this movement engine isn't isolated, right? It's highly networked with those survival pathways we just talked about.
Absolutely. And this really speaks to the integrated nature of the cancer network. ARP23 activity is often ramped up by regulatory proteins like RAC1 and CDC42. And if you remember from the first section, those are the same regulatory proteins that interact directly with the master survival pathway, PI3K.
So a signal to survive is also a signal to move.
They're constantly talking and reinforcing each other. A signal to survive is also a signal to move. Furthermore, ARP23 relies on the waving and WASPA complexes to sense chemoattractants, which means the tumors can actively smell and sprint toward new blood vessels or lymphatic nodes.
So it's not just random movement, it's highly directed invasion using self-generated force.
Precisely. It allows cancer cells to invade the basement membrane without always needing matrix metalloproteinases, the enzymes that dissolve tissue. They can just physically push through. But here is the major clinical twist, the real kicker that reveals this masterclass in operational stealth. ARP23 is essential for the initial invasion and the crawl toward the blood vessels. But once metastasis is complete, once the cancer cell arrives at the distant organ, like the liver or the lung, ARP23 shuts off.
Wait, why would it shut off the movement engine once it arrives? That seems counterintuitive.
Because if the cell is constantly crawling, it can't settle down and establish a new colony. To successfully colonize a distant organ, it needs to revert slightly from that aggressive ninja cell back to a more fixed, epithelial-like state, a process called mesenchymal epithelial transition, or MET-T.
So by shutting off ARP23, it can settle down quietly.
It stops its aggressive movement, settles down quietly, and becomes nearly undetectable during this crucial settlement and initial growth phase. It makes these new distant metastases so much harder to catch clinically. It's an elegant, horrifying survival strategy.
That truly highlights the layered complexity. The cell knows when to be aggressive and when to be silent. Okay, so once a new or primary tumor is established, it still faces the major hurdle of securing resources, especially when growth outpaces the blood supply. This takes us to the mechanisms of hypoxia and angiogenesis.
Right. When tumor growth accelerates rapidly, the cells in the core of the tumor, they run out of oxygen. This creates a low oxygen environment, or hypoxia. And cancer has learned not just to survive this stress, but to exploit it.
How so?
When oxygen levels drop, the HIF-1 pathway is activated. And HIF-1 acts as the signal that the entire tumor needs new resources, and it needs them immediately. It is the cellular distress call.
And that distress call culminates in flipping the angiogenesis switch.
That's exactly right. The core job of the HIF-1 pathway is to command the release of VEGF, vascular endothelial growth factor. VEGF acts as a powerful molecular magnet, forcing the surrounding normal blood vessels to sprout new branches and snake their way into the tumor mass.
So the tumor is literally forcing the body to build it a custom life support system.
New blood vessels to deliver oxygen and nutrients. It's a mechanism where cancer exploits its own starvation signal to guarantee its survival.
And what's interesting here is that those new blood vessels are often leaky and poorly formed, which actually creates an even more stressed, hypoxic environment.
Which reinforces the HIF-1 signal. It's a self-feeding loop of chaos. The tumor doesn't need perfect vasculature, it just needs more vasculature, regardless of its poor quality, to keep the core growth signals alive.
We've covered how cancer starts, grows, and crawls. Now we need to look under the hood at its internal factory settings, its energy generation, and its master genetic instruction manual. This is where metabolism and epigenetics get utterly chaotic. We're moving into section 3.
Let's start with the fundamental metabolic flip, the road power plant, mitochondrial rewiring. Most healthy cells use mitochondria to efficiently burn glucose with oxygen. It's a process called oxidative phosphorylation.
Right, which produces massive amounts of clean energy.
Exactly. Cancer flips this script dramatically, adopting the classic metabolic shift known as the Warburg effect.
That means switching to glycolysis compared to mitochondrial respiration. Why would cancer, which needs massive resources, choose a less efficient energy source?
The purpose of the Warburg effect is not maximum energy efficiency. It is maximum cell-building efficiency.
Okay, explain that.
Glycolysis processes glucose quickly in the cell's cytoplasm, and in doing so, it spits out these crucial raw material intermediates molecules that are diverted from energy production to be used as starting blocks for synthesizing lipids, nucleic acid, so DNA and RNA, and amino acids.
So it's less about generating clean ATP for energy.
And more about running a high-speed construction factory, producing every component necessary for rapidly assembling new cells. The lactate it excretes is just the byproduct of this hyperfast construction process.
So it's not wasting energy. It's aggressively prioritizing building materials for proliferation. And to ensure it gets all the fuel, it employs a highly specific, very sneaky trick regarding glucose intake.
It's pure glucose hogging. It's a territorial move. Cancer sticks the enzyme hexokinase directly onto the outer mitochondrial membrane.
And hexokinase is the first step in processing glucose.
Right. By placing it directly on the mitochondrial membrane, it essentially creates a private toll booth. It intercepts and modifies the glucose molecule right as it enters the cell, ensuring the cancer cell gets access to the fuel before any other normal cellular process can use it. It starves the neighboring healthy cells.
But despite this clear preference for the fast-lane glycolysis, the sources note that cancer still uses mitochondria, just strategically. It hasn't fully abandoned the power plant.
It's all about tactical flexibility. Mitochondria are used specifically for high-demand synthetic purposes. For instance, they crank out massive amounts of aspartate, which is absolutely crucial for synthesizing new DNA bases. Without that aspartate boost, rapid DNA replication would slow down.
And there's another surprise, right? With ROS.
Yeah, this is a major one. Cancer intentionally pumps out low levels of reactive oxygen species, ROS. Normally, ROS causes damage, but here it's a signaling molecule. It's used to keep the HIF-1 pathway lit, the one we just discussed that screams for new blood vessels, even if oxygen levels aren't critically low. It maintains that perpetual state of emergency to justify its growth. It's structurally similar to an essential molecule used by the cell's agenetic machinery to regulate gene expression. So by flooding the cell with 2-HG, the cancer cell essentially throws a wrench into the epigenetic clockwork.
And what's the result of throwing that wrench in? What does it do?
It incorrectly flips or silences genes all across the genome by inhibiting DNA and histone demethylases. It locks the cell into a permanent, highly aggressive tumor state. It prevents differentiation and promotes stemness. You can think of it as permanently saving the tumor's malignant settings.
Wow. And there are also smaller, but still highly effective tricks for regulatory control, like summoylation and the use of what the sources call microRNA crews.
Yeah, so summoylation involves adding these tiny tags, small ubiquitin-like modifiers, to key proteins. This is a stability hack. It stabilizes central oncogenes like HIF, our hypoxia switch, or c-MYC, our master growth regulator, and it prevents their normal degradation by the cell's recycling systems.
Well, they just hang around longer, maximizing their impact.
Exactly. It's like gluing the on switch in place, making degradation impossible without rewriting the entire cellular recycling manual.
And the microRNA crews, how do they work?
They function as highly efficient little silencers. These small RNA molecules, like miR-21, they don't code for proteins, but they patrol the cell and bind to messenger RNAs, silencing their production. Cancer leverages these, allowing a single microRNA to silence multiple tumor suppressors simultaneously on the cheap.
It's an extra highly efficient layer of control.
Without needing to physically mutate multiple genes, yes.
Let's shift to the spatial and physical tricks the cancer employs to gain an advantage in its immediate environment, starting with the breakdown of the Hippo pathway.
The Hippo pathway normally acts as the cell's density monitor. It tells cells, hey, stop stacking up, you're too crowded. It prevents cells from overgrowing and keeps organ size regulated. When cancer switches this pathway off, proteins like YAP and TAZ are released from regulation, and they just go nuts.
And what do YAP-Z tell the cell to do when they go nuts?
They tell the cells to ignore density boundaries, to stack higher, to ignore their neighbors, and to proliferate aggressively. This often leads to the formation of organoids, mini-organs within the tumor mass. This loss of spatial discipline is a hallmark we see frequently in liver, breast, and gut cancers.
It allows the tumor to grow beyond its normal structural boundaries.
Exactly.
And then there's the incredible manipulation of local chemistry, which I found fascinating, the proton reversal tactic.
This is a simultaneous defense mechanism and an invasion tool. Cancer relies on the fact that its high-speed glycolysis spits out massive amounts of acid, lactate. To counteract the toxicity of that acid internally, cancer pumps protons out of the cell.
And this achieves two things, right?
Two things. First, it maintains a higher optimal pH inside the cancer cell, which is perfect for their rogue metabolism. Second, it makes the external environment, the space between cells, extremely acidic.
And that acidity must do serious damage to the surrounding tissue.
It does. That acidic environment helps the tumor dissolve surrounding tissue, which aids metastasis. But crucially, immune cells like T cells and natural killer cells are incredibly sensitive to low pH. So this creates a literal chemical shield around the tumor that helps it dodge immune attacks.
It's optimizing its factory while creating a hostile battlefield for the immune system.
At the same time.
And lastly, to complete this picture of metabolic and physical mastery, we need to detail a few specialized survival strategies for apoptosis resistance and glutamine addiction.
Right, so apoptosis is a regulated, iron-dependent form of cell death driven by lipid peroxides, basically oxidative stress that fries the cell membrane. It's a defense mechanism the body uses against tumors.
Okay, so how does cancer get around it?
It has a specific cheat code. It hoards iron-binding proteins, especially one called GPX4. And GPX4 neutralizes those lipid peroxides, preventing the cell from frying itself. It's a key survival mechanism under metabolic stress.
And the glutamine addiction reinforces the metabolic chaos we discussed with glycolysis.
Yes. While they rely on glucose for raw materials, many aggressive cancers become addicted to glutamine, which they process using the enzyme GLS1. Glutamine is essential because it feeds carbon and nitrogen into the Krebs cycle, supporting their high metabolic rate and providing building blocks for proliferation, even if glucose is temporarily scarce.
Which makes GLS1 a highly promising drug target.
Exactly. If you can cut off the glutamine, you can severely cripple these addicted tumors.
So, cancer has perfected its internal machinery, its movement, and its energy supply. Now it turns its attention outward, starting with the most formidable threat, the immune system. Section 4 covers the complex ways cancer manipulates its immediate surroundings and even distant partners, like gut bacteria.
And the first, and now perhaps most famous, strategy is the invisibility cloak, provided by the immune checkpoint gang. We all know the breakthrough of immunotherapy targeting PD-L1.
Right.
PD-L1 overexpression on the tumor cell signals T cells to just chill out, providing that literal invisibility cloak by stopping the attack before it even starts.
But the sources remind us that PD-L1 is just the tip of a large coordinated iceberg. It's an entire gang of checkpoint markers working in concert.
That's absolutely right. The immune system uses many inhibitory receptors to make sure it doesn't accidentally attack healthy tissue. Cancer has learned to exploit the full lineup: CTLA4, LAG3, TID. They're all key players.
And they all deliver inhibitory signals to the T cells.
They mute the immune response, making the T cell sluggish or forcing it into exhaustion. And the strategy for the future involves targeting these multiple checkpoints simultaneously, a combination immunotherapy approach.
Beyond just muting the T cells, how does cancer actively create an environment that promotes its survival and supports this evasion?
It relies on sustained chronic inflammation, and it leverages the STAT3 inflammation loop. Cancer uses this pathway to create a low-grade, persistent inflammatory state that, counterintuitively, actually promotes the tumor's thriving.
So not the acute, intense inflammation that signals an immune alarm.
No, it's a sustained, smoldering fire that helps the cancer grow, promotes cell survival, and constantly signals to immune cells to remain suppressed or to differentiate into pro-tumor subtypes.
This naturally leads us to a stunning discovery of the last few years, cancer's microbial army. The idea that a distant partner, the gut microbiome, is actively collaborating with the tumor.
It is a remarkable and, frankly, deeply concerning piece of biological sabotage. Cancer actively recruits and favors specific species of gut bacteria like Fusobacterium nucleatum or certain bacteroid strains. These are often opportunistic pathogens that thrive in the compromised environment created by cancer.
And how do these bacteria actively help the cancer?
They function as a chemical sabotage crew. They start pumping out toxins that directly poke holes in the host cell's DNA or flip internal growth switches like NFB, which, if you remember from Section 1, promotes endless pro-tumor inflammation and blocks apoptosis.
So the bacteria are chemically enabling the cancer's core survival pathways.
They're feeding the fire that the cancer lit.
Exactly.
And they also help with chemical conversion, essential for tumor growth. Gut bugs convert relatively harmless bile acids produced by the liver into much nastier secondary bile acids, like deoxycholic acid. And these molecules are highly toxic and directly fuel the formation of polyps and tumors, particularly in the colon.
It's a localized, highly potent chemical attack on the host tissue. And the signal gets even sneakier through leakage, linking the gut back to that STAT3 fire we talked about.
Yeah, so cancer and its microbial allies, they compromise the integrity of the intestinal lining. This allows leakage of bacterial components, such as LPS, into the bloodstream.
Lipopolysaccharide.
Right. And this LPS sustains that low-grade STAT3 fire we just mentioned. So a piece of a bacterium that should stay locked in the gut is leaking out, and that leakage is sustaining the exact inflammatory state that keeps the T cells from attacking the tumor. It's an elegant, horrifying, self-sustaining loop.
And in colorectal cancer specifically, the collaboration becomes physical, doesn't it?
Yes. In that specific instance, bacteria like Fusobacterium actually move into the tumor itself, taking up residence and forming protective biofilms. And once they're inside, they churn out enzymes that help the cancer cells spread and further the metastatic process. They're essentially biological co-conspirators residing within the tumor's fortress.
Finally, in this section, let's discuss the methods cancer uses for distant communication, hijacking the body's messaging service via exosomes and manipulating the body's physical defenses via nets.
Exosomes are the cell's universal postal system. They're these tiny lipid bubbles packed with RNA and proteins that cancer cells spit out like molecular messages in a bottle. They travel through the bloodstream and the lymphatic system, delivering instructions to distant cells far from the primary tumor.
This is critical for preparing for metastasis, isn't it? What kind of instructions are they carrying?
They have three main devastating functions. First, they reprogram distant cells. They turn healthy supportive cells, like fibroblasts, into highly collaborative cancer-associated fibroblasts, or CCAFs.
And they start building a supportive scaffolding for the tumor.
They do. Second, they prepare the pre-metastatic niche by rolling out the cellular red carpet, ensuring the environment is perfect for a new tumor cell to settle down when it arrives. They deposit adhesion molecules, growth factors.
And the third function involves the immune system, turning protectors into saboteurs.
Yes. They turn certain immune cells, like neutrophils, into immunosuppressive N2 goons, a type of cell that actively suppresses T-cell activity. This reinforces the invisibility cloak far away from the primary site, making sure the new metastasis is ignored.
And how do neutrophil extracellular traps, the nets, figure into this physical defense mechanism?
Nets are a defense mechanism gone terribly wrong. Neutrophils are white blood cells that fight infection. To catch large pathogens, they sometimes explode their DNA and proteins into these sticky extracellular webs. Those are the nets.
Okay.
Cancer tricks neutrophils into doing this unnecessarily. Normally, these traps catch bacteria, but here, cancer uses them as a physical shield and a travel aid. The webs literally shield the cancer cells from the sheer stress of the blood flow and aid the cancers crawling into and out of blood vessels, facilitating metastasis.
So it's using the body's own defense system as a launch pad for spread. Everything we've covered so far sets the foundation, but to truly understand the 2025 survival playbook, we have to dive into the deep, dark corners, the highly sophisticated molecular and physical tricks that complete this portrait of the monster. This section is about cutting-edge cell physics and highly nuanced genomic manipulation that feel almost futuristic.
These are the hacks that give cancer its incredible adaptability. Let's start with genomic and RNA manipulation, specifically the idea of making messenger RNA mRNA live forever using alternative polyadenylation and 3-UTR shortening.
Okay, let's unpack this slowly. Why does shorter mRNA mean eternal life for the oncogene? That seems counterintuitive.
So when a cell creates a messenger RNA, the recipe for a protein, that recipe has this functional tail called the 3-UTR, the 3' untranslated region. And this tail contains binding sites where regulatory microRNAs, or little silencer crews from section 3, normally grab the mRNA and signal for its degradation.
So the UTR acts like a biological fuse, ensuring you don't make too much protein.
Precisely. It's the degradation signal. Cancer uses this sophisticated trick called alternative polyadenylation to create oncogene mRNAs that are shorter. They lop off most of that regulatory 3' UTR tail. By removing this segment, they literally remove the target site for the regulatory microRNAs.
So the silencers can't grab on.
They can't. And this ensures pro-growth messages like NYC and cyclin D1 live forever and are translated into massive amounts of protein indefinitely. It's a genius stability hack that bypasses the cell's natural recycling rules.
That is a fundamental manipulation. Moving on, we have RNA methylation rewiring, specifically the M6A and M1A tags. This sounds like epigenetics, but applied not to DNA, but to the RNA message itself.
That's exactly what it is. These M6A and M1A methylation tags fundamentally change how quickly pro-growth mRNAs are read and translated into protein. It's not about changing the gene itself, but changing the speed and the longevity of the protein factory.
So you have enzymes that place the tag and readers that see it.
Yeah, we're talking about enzymes like METL3 and METL14, which place the M6A tag on the RNA, and then YTHDF readers that recognize that tag and make the transcription process dramatically faster.
It sounds like they are creating a fast-pass lane for their own recipes.
Absolutely. The source even notes that the enzymes responsible for putting these tags on, METL3 and METL14, are now considered near universal oncogenes because they accelerate proliferation across so many tumor types. They are a universal speed control switch that cancer loves to flip to maximum.
And speaking of editing, cancer also messes with the cell's internal editor, the spliceosome.
The spliceosome is the highly complex cellular machinery responsible for editing the initial RNA transcript. It cuts out the junk sequences, the introns, and splices the useful pieces, the exons, together to form the final functional protein recipe.
So mutations in that machinery must cause chaos.
Mass confusion. Mutations in core spliceosome components like SF3B1 or U2AF1 lead to the creation of entire new protein isoforms, alternative versions of proteins that only the cancer cell can use for survival and growth.
So it's proprietary software that healthy cells can't run.
Exactly. For example, a shortened version of a protein might have lost its inhibitory domain, turning it into a hyperactive oncogene. It's a way for cancer to custom design its own toolkit from existing human blueprints.
And finally, a truly highly technical DNA structural hack, G-quadruplex exploitation. This sounds like cellular physics manipulation.
This is literally a physics hack on the DNA structure itself. While DNA is usually a double helix, certain guanine-rich regions in the promoter areas of genes can fold into these highly stable, four-stranded structures called G-quadruplexes.
Okay.
And cancer stabilizes these structures, often with specific proteins, in the promoter regions of key oncogenes like MYC and KRAS.
And why stabilize them? What's the benefit?
By stabilizing them, it physically prevents the growth genes from ever shutting off, regardless of cellulase link. The quadruple helix structure acts as a physical block that ensures the transcription machinery can dock there permanently, maximizing the output of those critical growth genes.
Moving from the purely molecular to the physical environment, the next set of tricks is about manipulating the cell's internal physics. Let's talk about the surprising concept of phase separation and biomolecular condensates.
This is where molecular biology meets thermodynamics. Some regulatory proteins, particularly those involved in aggressive tumors like sarcomas fused in WSR1, can undergo phase separation. This means they physically separate from the surrounding cellular fluid and turn into liquid-like droplets or condensates inside the nucleus.
Liquid droplets inside what should be a solid, structured nucleus. That's difficult to picture.
Think of it like a specialized, dedicated 3D printer running inside the general factory floor. These droplets act as cheating rooms, where oncogenes are concentrated. By sequestering the transcription machinery, the relevant DNA, and the transcripts all inside this tiny concentrated liquid environment, transcription just runs wild.
It's a localized factory running outside the usual rules.
A highly efficient, localized factory running outside the usual regulatory controls of the cell. It massively boosts the production of cancer-promoting proteins far beyond normal levels.
Next, let's dive into mechanical transduction. The sources suggest that the physical stiffness of the tissue itself can flip the malignancy switch. That implies cancer is not just chemistry, it's physics.
This is a major insight that's really driving research right now. The tumor actively makes the surrounding tissue rock hard. It creates a stiff, fibrotic extracellular matrix, or ECM. And this physical stiffness alone is enough to transmit powerful signals back into the cell via specialized surface receptors called integrins.
And that physical force is transduced internally.
Yes, and it activates the YAP pathway, the same one involved in the Hippo breakdown we discussed earlier.
So the physical environment literally dictates the cell's genetic behavior.
It does. The stiff matrix supplies tension, and that tension is interpreted by the cell as an unrelenting grow and divide signal. It drives malignancy, invasion, and chemotherapy resistance. If you change the stiffness, you change the cell's fate. This connection between the mechanical world and the molecular world is critical, and we'll definitely revisit it in our final thought.
We also see evidence of cancer sabotaging the body's fundamental rhythm, the circadian clock sabotage.
Every cell runs on a 24-hour clock governed by feedback loops involving PER and CRY proteins. This clock regulates when cells sleep, divide, and repair DNA. In aggressive cancers like breast, liver, and leukemia, this natural loop is disrupted.
Why? What's the benefit?
By breaking the clock, the cells are forced to divide 247 instead of resting at night. This maximizes proliferation speed and also disrupts DNA repair mechanisms, making the cancer cells more mutated and aggressive.
And there's a fascinating immune evasion trick that involves viral mimicry.
Our genome contains dormant endogenous retroviruses. These are ancient viral DNA sequences that have been embedded in our DNA for millions of years. Some cancers turn these sequences back on.
And that makes the cell think it's infected.
It tricks the cell into thinking it's infected by a foreign pathogen. This keeps the S-sting pathway chronically lit. And while S-sting normally signals the immune system to fight viruses, in the context of cancer, this chronic low-grade signaling often promotes metastasis and survival. It acts as another inflammatory signal that helps the cancer spread rather than fight it.
Finally, we need to circle back and look at two survival details: lipid droplet hoarding and the betrayal of senescence.
Lipid droplet hoarding is a very shrewd resource strategy. Cancer, knowing that chemotherapy and nutrient deprivation are likely, stores massive amounts of fat inside the cell in protective lipid droplets. These serve as an internal energy bunker, ensuring survival during periods of stress. It's stockpiling fuel for the siege.
And the SISP betrayal is another layer of environmental manipulation, using cells that should have simply died.
Senescent cells are old cells that have stopped dividing and should be cleared by the immune system, but sometimes they refuse to die. And these senescent cells, they vomit out a toxic mix of molecules, the senescence-associated secretory phenotype, or SASP.
And that mix includes growth factors.
Potent growth factors like IL-6 and VEGF, and tissue-dissolving enzymes like MMPs. They actively feed the young proliferating tumor next door, creating a literal cocktail that accelerates the growth and invasion of the cancer. It's the ultimate act of betrayal by cells that should be retired.